today's episode of Myeloma Crowd Radio, a show that connects patients with myeloma researchers. I'm your host, Jenny Ahlstrom, and we'd like to thank our episode sponsor, Karyopharm Therapeutics, for their support of Myeloma Crowd Radio and this program. Now, before we get started on today's show, I just want to make a short reminder announcement about recent updates inside of HealthTree. You may have heard in this last week's show, but it bears repeating. We now have new notifications and messages inside of HealthTree. So if somebody reaches out to you for a twin request or comments on one of your posts, you're going to see a little bell at the top of the screen with a red circle in it with your number of messages that you have. You'll also see twin messages in the chat icon, so you can chat back and forth with your twins. And in the future, you'll also be able to chat with people directly back and forth, like direct messaging inside of the forums. So we are listening to your feedback, and we're improving HealthTree to provide more functionality to you over time. We also have a new feature in development, and we encourage you to enter your prior treatments and side effects to get ready for this new feature. I'm still going to keep you in suspense, but I know you're going to love it. So we have a team ready at all times to help you enter your information, and we can give you electronic ways to do it yourself, or we can do it for you if you prefer. And now on to today's program. We hear about personalized medicine in cancer, but really what does it mean for us as myeloma patients? Top myeloma expert Jonathan Kaufman of Emory University joins us today to discuss what could be considered the first true personalized medicine for myeloma patients who have the 1114 translocation. And we're very excited to learn more about this. So Dr. Kaufman, welcome to the program.
Great, Jenny. Thanks so much for having me, and looking forward to meeting with the myeloma crowd today. So it's a lot of fun for me. I've never done your radio version before, and I'm looking forward to sometime in the future where we get to all do this in person again.
Oh, I know. Well, yeah. Dr. Kaufman participated in one of our roundtables in Atlanta, and you were just fantastic. So we're just so appreciative that you're joining us for this particular topic today. So before we get started, let me introduce you. Jonathan L. Kaufman is Associate Professor and serves as Associate Vice Chair and is Director of the Division of Hematology in the Department of Hematology and Medical Oncology at Emory University School of Medicine, which is also part of the Winship Cancer Institute. He's board certified in hematology and medical oncology. Dr. Kaufman his practice includes treating myeloma and amyloidosis patients at Emory University. And as a network physician, he started practicing at Emory in 2005. He's also an active clinical and translational researcher in the fields of myeloma, amyloidosis, and bone marrow transplant. He's a member of the Discovery and Developmental Therapeutics Research Program at the Winship Cancer Institute. He has memberships with ASCO and ASH, and his awards include Atlanta's Magazine Top Doctor in 2015, ASBMT New Investigator Award, an ASCO and AACR Methods in Clinical Cancer Research Award, and additional awards. His work has been published in journals such as the Journal of Clinical Oncology, Science Signaling, Blood, Leukemia, Journal of Immunology, and many, many others. He has presented his clinical research on multiple myeloma at invited lectures as well as national and international hematology and oncology conferences. So, Dr. Kaufman, I know you've been heavily involved in Venetoclax clinical trials. So maybe we just want to begin with a broad overview for what is Venetoclax and uses for Venetoclax in myeloma patients in general.
Great. Thank you so much again. I think so Venetoclax is a drug that targets a protein called BCL2. And I think it's important let me just get to I find a good way to talk about this is understanding what BCL2 is in the myeloma cell. And I'm going to spend a few minutes talking about really basic myeloma biology and really basic cancer biology. One of the things that we know about cancer cells, every cancer cell, including in every myeloma cell, is that they are very sick. And the cell is sick and it wants to die because there are so many signals within the cancer cell. The cell itself wants to die. But in order for it to turn into cancer, this very sick cell has to develop a strategy to prevent itself from dying. And so it creates or overexpresses or modifies in some way all of these anti-death proteins that keep the cell alive. And there's several anti-death proteins. One of them is the target of Venetoclax called BCL2, and then there's another one called MCL1, and then there's another one called BCLXL. And so all of these proteins are there to keep the cell from dying. And they work in a very complicated and coordinated fashion with each other and with similar proteins to keep the cell alive. And almost every myeloma cell is in some way dependent on MCL1 to stay alive. And there's currently a lot of clinical investigation, clinical trials, research into drugs that target MCL1. But they're not really far in development at all, and there's really very little clinical data about MCL1 inhibitors. But BCL2 in a subset of patients is the protein that is really important in keeping the cell alive. And so Venetoclax is a drug that specifically targets BCL2. And if you remove, it's like the Venetoclax basically removes the guardian of the cancer cell and allows the cancer cell to die because that's what it wants to do. So it's really amazing technology in terms of identifying these proteins that sit in this perfect little pocket to stop this protein called BCL2 from working.
Okay.
So Venetoclax is it's an oral medication. It's a BCL2 inhibitor, and it's approved for several cancers. It's approved for acute myeloid leukemia. It's approved for chronic lymphocytic leukemia, and it's very effective. I'm very lucky to work here with one of my scientific colleagues, a scientist, Larry Boyes, who I would argue is one of the top 2-3 world's experts in understanding the medical term is apoptosis, or the other name of it is cell death, but really understanding cell death in myeloma. And we published an article in 2011 in the journal Blood, where we looked at a drug like Venetoclax, and we were able to identify and really understand the reason why certain cells respond to Venetoclax and certain don't. And so I'll just quote you this quote. It says, "These data have several clinical implications, such as selecting the correct subset of patients to treat with Venetoclax, as well as searching for agents that may synergize with this drug." So we knew almost 10 years ago that Venetoclax was going to be a drug that was only going to work in a subset of patients.
Okay.
So the obvious question then becomes, well, who is that subset of patients? Isn't that what you were just going to ask?
Yeah. Well, my question was actually about, like, in other cancers, you talked about AML and CLL.
Yeah.
Is it effective just broadly or just for a subset of patients in those cancers as well? Because that would give you an indication.
That's a great question. That's a great question. So you know how I mentioned that in the beginning that almost all myeloma cells are dependent on the other protein, the MCL1, and it's the exact opposite in leukemia, in AML and CLL, acute myeloid leukemia and chronic lymphocytic leukemia. They're almost all dependent on BCL2. So they don't need to do subsets because they're the exact opposite.
Interesting. Are the MCL1 inhibitors the ones that are being looked at in the 1Q gain, or no? Do I have that wrong?
Yeah, that is true. So the reason why people think that MCL1 inhibitors might work more in 1Q gain is because MCL1 is on 1Q, and so it makes a lot of sense. If you have more of 1Q, you'll have more of MCL1, and in theory, if you have more of MCL1, then that cell should be more sensitive to MCL1 inhibitors.
So interesting. Okay, I didn't know that, the difference between the leukemias and myeloma.
Yeah. So we were in. So then we went. So after that initial paper, we went and queried a whole bunch of cell lines with the drug, and then we have a protocol where we take patient samples, and then we look and we compare and we test the patient samples. And what we found is that there was the cell lines that harbored the 1114 translocation, and the patients who had the 1114 translocation were much more likely to respond in the laboratory than other cell lines and other cytogenetic abnormalities.
Mm-hmm. And why is that? Like, what is it about 1114 that just has more BCL2 in it?
This is a great question. Why? So just before sort of going into why the T1114s are more, just to again sort of understanding what T1114 is. And again, you guys probably all already know this, but I'll just go through it again. We have 23 paired chromosomes in each cell, and they're numbered 1 through 22, and then either XX or XY. And we know the different sizes of these chromosomes, and that's how we label them. And so what happens in the 1114 translocation is that a portion of the 11th chromosome gets mixed and matched with a portion of the 14th chromosome. And that's the 1114 translocation. And and again, as a reminder, myeloma is cancer plasma cells, and plasma cells' job in life is to make antibodies. And the gene for making antibodies is on the 14th chromosome, and so that's why when we see all these cytogenetic abnormalities in myeloma, you'll see 14 come up over and over again, because 14 is where the gene that makes the antibody comes. And so again, we're going back to we're going back to basic cancer biology. And what makes a cell turn into cancer is if you put in a gene that makes the cancer grow or prevents the cancer from dying, and then you put it next to right next to the portion of the gene that says, I'm going to make you all the time. So instead of all the time making an antibody, the gene is all the time making a protein that tells the cell to grow, or tells the cell to not die, or tells the cell that you can be resistant to chemotherapy, or whatever it is that makes the cell go from normal to cancer.
And I've heard somebody talk about chromosome 14 as like the promoter gene, like you're talking about. Like,
right,
okay,
that's exactly right.
We've got the signal, and now it's going to go. And then what does 11 do?
So that's a great question. What's on 11 is a protein called cyclin D1, and cyclin D1 is involved in telling the cell to grow more. And so the very first question that we asked is, does the sensitivity to Venetoclax, to BCL2 inhibition in 1114, have anything to do with cyclin D1? And we learned very early that it has nothing to do with the actual translocation. So the sensitivity of Venetoclax in 1114 myeloma has nothing to do with the product of that translocation. And so then we had to dig deeper and understand more. And again, that's where this work we've done in the laboratory, and what we found in the laboratory is that it's these 1114 cells that are much more likely to be associated with BCL2 as the protein that's protecting the cell from dying than any other than other myeloma cells. And the other thing that's really interesting about 1114 myeloma, which we've actually known for a long time, is that 1114 myeloma is a little bit different clinically. The cells look, a classic 1114 myeloma cell is going to look different than another myeloma cell. And it's interesting, and I think you Jenny, you might have picked up on this. The 1114 myeloma cell looks a little bit more like a lymphoma or a CLL, just a little bit. And the proteins that are on the 1114 myeloma cell are more likely to be the lymphoid or the lymphoma-like proteins. And so we think that one of the reasons that a certain subset of 1114 myelomas are more likely to respond is because they act a little bit more like a lymphoma than a pure myeloma.
Well, that's
so I just want to be very clear. It's still myeloma. It's still 100% myeloma,
right,
but it acts just a little bit differently.
Have other lymphoma or leukemia drugs been reviewed for 1114 patients?
Well, yes, and there doesn't seem to be. So it's not enough. It's not enough like a lymphoma that lymphoma drugs would work. If you think about drugs like rituximab or ibrutinib and things like that, those have been tried and not to be effective. But that's a great question. The question is, can we? But it's really, so it's not enough of a lymphoma to do that, but it's enough to make these cells potentially sensitive to drugs like BCL2, drugs like Venetoclax that target BCL2.
Interesting. And when you think about 1114, I know there are different types of heavy and light chain myelomas like IgG or IgA or IgM or whatever. And then 1114 is typically considered lower risk. Do you want to go into any of that? Are there certain types that are found in 1114 mostly, or?
That's a really important question. So there is probably there's a lot of data out there asking the question, how do patients with 1114 do? And historically, patients with 1114 translocation were not considered high-risk disease, were considered standard risk disease. And then with the advent of drugs like all the new drugs we've had, I say new, but now we're getting on almost 20 years old now. All the proteasome inhibitors like Velcade and the IMiDs like Revlimid, and those drugs really improve the outcome of patients with myeloma. And it's interesting, if you look at our data, we recently published a series of 1,000 consecutive patients treated in a uniform way. And then we asked the question, how did the 1114 patients do? And the answer is that the 1114 patients in our large data set, on average, didn't do as well as other patients with standard risk disease. And so we really think that there's a huge need for new drugs, specifically in 1114 myeloma, because they don't benefit as much from what I call the pure myeloma drugs, drugs like Velcade and Revlimid. And it's interesting. The other thing that's clinically about 1114 myeloma is that these are the patients with 1114 myeloma are more likely to have light chain only disease. They're more likely to have non-secretory disease. They're more likely to have plasma cell leukemia. So there's a whole variation in 1114 that makes it a little bit unique compared to other myelomas.
And I know non-secretory or secretory and PCL, I mean, those are considered kind of more advanced and trickier diseases to work with.
Yeah, I mean, certainly the plasma cell leukemia, but if you look at plasma cell leukemia patients, you can really divide them between if they have 1114 and if they don't have 1114. And again, we have a publication that demonstrates that 1114 myeloma patients who present with plasma cell leukemia are much more likely to live a long time than those patients with plasma cell leukemia that don't have 1114.
Oh, interesting. Okay, so it kind of makes it better for them.
Yeah.
Can we talk about bone damage as well for 1114 patients? Do you see a lot of bone damage typically?
So in general, this is not, you can, just like any myeloma can have bone disease, but it's not considered one of those that is where you have a high bone damage burden with 1114. I'm not saying it can't happen, but it's not the classic feature of 1114 myeloma.
Oh, that's interesting to know because, yeah, I know some of the myelomas are like slower growing, but a lot more bone damage, and some are very, limited bone damage and all that. That's so interesting.
Right.
So when you think about, I mean, we talked kind of at the intro about personalized medicine, and this is really, it's kind of confusing in my mind because you think, I've heard lately from different myeloma researchers that, at diagnosis you actually have like thousands of mutations, and then after treatment you have 10x those thousands of mutations. So targeting things directly with one type of drug or, like a BRAF inhibitor or something like that might be challenging, but it doesn't seem to be the case in the 1114, so it seems to be really pretty ideal.
Yeah, I think so this isn't really, this is different than a mutation.
Okay.
And I think we're obviously very aggressively trying to develop personalized therapies based on mutations in myeloma, but myeloma is not a simple disease. And whereas there are, and then where the diseases where you can find that one mutation that controls everything, we just haven't found that yet broadly in myeloma, and we're continuing to work to see if we can find it.
Mm-hmm.
And so the thing about 1114 is that the product of 1114 isn't our target. But 1114 is the marker that this is a cell with a different underlying biology. And so that's the difference.
That makes a lot of sense. Okay, well, thank you for clarifying that because it just, I'm seeing so much like efficacy in some of this particular targeted approach. And then the other part of personalized medicine just seems more challenging to me.
Yeah, and again, it's again the 1114. I would say so. It's really what we call a biomarker. It's the marker that there's likely increased sensitivity to the drug, as opposed to the target of the drug.
Mm-hmm. Okay, that makes a lot of sense.
Go
ahead.
Go ahead.
Oh,
I was going to say it's sort of then a natural time to start talking about the drug and the targeting and
for sure.
all that kind of stuff.
Yep, let's do that.
So there was, just like we do in all of new drug development in myeloma, we had to do what's called a phase 1 study. And when you do a phase 1 study, you often treat patients where there's not other treatments available. And so this was a phase 1 study, and it was originally done in the original phase 1 study, which has now been published, was done for any myeloma patient who had relapsed disease and really didn't have any other options. Now, as I said, we've known for a decade here, and we've been saying to anybody who would listen to us, that what we really need to do is target the subset of patients. And so the whole study had 66 patients. You would predict that around 15% of the patients in any trial will have the 1114 translocation. In this trial, of the 66 patients who went on the trial, 30 of them had the 1114 translocation.
That's almost half.
And
wow.
Almost half. And the reason that happened is because our center and a handful, and then once people started seeing the data that came from our center and a handful of other investigators, started going away from non-1114 patients and towards 1114 patients. And so it ended up being about half of the trial had 1114. So if you look at, and I think if you look at the whole trial, the response rate was about 20%. But then if you look at the difference between the 30 patients who were treated who had 1114 versus the 36 patients who did not have 1114, the overall response rate, and this is single agent venetoclax, no dexamethasone, no combination, no nothing, the response rate was 40%.
That's much higher than normal.
Yeah, I mean, so if you think about response rates for single agents, Velcade in the phase 1 study was 30%. Single agent Revlimid was 30%. Single agent daratumumab was 30%. So it's because we were able to understand, because we understood the biology better, we were able to increase. We were able to get a response rate of 40%. And if you looked at the patient who didn't have 1114, the response rate was 6%, which is also consistent with our data that suggests there are some and non-1114s that we predict would respond to venetoclax. Unfortunately, we don't have that biomarker completely nailed down, and there are some thoughts about that. And the largest thought is those patients who don't have 1114 but have very high BCL2 levels, that those patients might respond to venetoclax. So I would say that's much less known and a very intensive area of activity. I mean, of investigation. And
how do you test?
We have.
Go ahead.
It's a protein that's in the cell, and there's different ways to test the level of the protein within the cell. But you have to, and so what we're doing, and we should be publishing shortly, is we've developed an assay that looks at the different proteins on the cell surface. And one of our physician scientists here, his name is Vikas Gupta, has developed an algorithm that also selects for both 1114 patients and a lot of non-1114 patients that are more likely to respond to venetoclax.
So fascinating. But you can find those.
Yeah, so,
I mean, that's amazing.
Yeah. So I think that's our future. So 1114 is good because you can get from a 6% response rate to a 40% response rate. But what you really want is to have a biomarker that's so good that even if it's a smaller number of patients, you have a 100% response rate.
Yeah. Yeah, definitely.
If we could predict, that's predictive medicine, which is you don't say, the doctor doesn't say to you, yeah, there's a 30% chance of this working, there's a 40% chance, there's an 80% chance. We say, you have this biomarker. Every single patient who takes this drug responds. That's precision medicine.
Yeah. That's where we as patients would like to see things go.
Yeah. I mean,
yeah, for sure. Before you take it.
Yeah. But that's essentially, that's Gleevec in CML, Yeah. All those, that maybe it's not 100%, maybe it's 98%. So that's what we'd like, that's where we'd like to be with our precision medicine.
Yeah, that's amazing.
Okay, so we got a 40% response rate, and said that this is great. This is a signal. This is real. So that, I think, and I will tell you, I remember going to meetings and saying, I just cannot believe how excited, because we had this one woman who was very young, and she was told there was nothing left to do, and she came to us. She had 11-14, and she went into a CR, and it's amazing. It's just really amazing.
Yeah.
So the next thing we want to do, and remember that opening statement I said, is that we got to figure out what the subset is, and we got to figure out what the best partners are. Again, Larry Boys in the lab, and Vikas Gupta in the lab, and Shannon Matulis, all scientists went back to the drawing board and asked the question, what's the best combination? And so we used all the standard combinations, and people might not believe me, but the best combination with Venetoclax was dexamethasone. Okay.
Oh, so interesting.
And the group of scientists able to figure out exactly why. And so it's funny because I have one of my colleagues says dexamethasone is like bacon; it makes everything better.
Mm-hmm. Yeah.
But
it is.
But in this situation, and then I also say about dexamethasone is always dexamethasone is both our friend and our enemy, and we have to figure out how to manage that at all times. And I think every myeloma patient knows that. But the question we had, so the reason why dexamethasone makes Venetoclax better is because it shifts more patients to being dependent on BCL2.
Oh, really? Okay.
Yeah. So we figured out we had this biology that we predicted, and it was the one drug that was more likely to make to make more patients sensitive, at least more patient cell lines sensitive to BCL2, to Venetoclax. And so then we did a trial with we sort of did two trials, and and the first trial we did, which was relatively earlier in the course of myeloma therapy, showed a 60% response rate. And then we did a trial where 90% of the patients were refractory to daratumumab, and in that trial, in combination with dexamethasone, and all these patients were refractory to dexamethasone, there was about a 50% response rate. And so we're really looking at very impressive outcomes, because if you look at drugs with patients with refractory myeloma like pomalidomide and dexamethasone, the response rate in that setting is 30%. And so we're looking at a 50 to 60% response rate in a patient population that is already refractory to a daratumumab combination.
Yeah. And it feels a little scary once you get refractory to through Dara, and you've used other things.
Yeah.
So.
Yeah. Okay. So, and that at this point, everybody asks the obvious question, which is, how come this is not available widely? Why is this still investigational? And and so there's been a lot of research of combining drugs, the proteasome inhibitors, with Venetoclax. And the rationale by using proteasome inhibitors is that proteasome inhibitors, block, in theory, they have a way of blocking MCL1. So if you stop MCL1 from being effective and you stop BCL2 from being effective, then you should have a very potent combination. And the preliminary research suggested that the combination of Venetoclax and Velcade and dexamethasone was effective. And then that led to the Bellini trial, and the Bellini trial was a large international trial that compared relapsed myeloma patients, and they had Venetoclax, Velcade, dexamethasone versus Velcade, dexamethasone. And the data came out that showed that the Venetoclax arm, if you take the whole population, did better than the non-Venetoclax arm. That's great. The problem was that there was an increased death rate in the patients who had Venetoclax versus the patients who didn't have Venetoclax. So despite the whole group doing better, keeping the myeloma from progressing, the whole group on average had much more toxicity, and that patients died.
And what about it was causing that?
Yeah, so
that halted that trial.
Halted the entire that halted the entire development of Venetoclax, and it's that data that is why Venetoclax is, that among other things is why Venetoclax is not available to us, all of us today, outside of the context of a clinical trial. And so there's a couple of things. If you it looked like there was, but remember that they took all comers. So if you just looked at about 10% of the patient population, consistent with what we'd expect, had 1114. These patients did not, at least in the very small numbers, did not appear to have a worse survival. But the numbers are so small that you can't tell for sure. And so, but the data really looks like the problem was in the non-1114 patients. And there's a couple hypotheses. Maybe there was an increased infection risk, and now all the studies are very conscientious about being on antimicrobials during treatment, and then making sure your vaccinations are up to date or getting up to date and things like that. And that really and so now all of the trials that are out there right now have stopped focusing on all myeloma patients and started solely focusing on the 1114 patients.
Well, let's talk about some of those trials, if you don't mind.
Yeah. So where are we now with trials? We presented at last year's American Society of Hematology conference the combination of Venetoclax plus daratumumab and dexamethasone looks to be this is in patients who had never had daratumumab before, looks like to be a very potent regimen. We now have a randomized study ongoing that looks at 2 different doses of Venetoclax with daratumumab versus Velcade plus daratumumab. So we have a clinical trial with a control to make sure that we don't see that change, that difference in survival, that decrease in survival. And so that we have, and that study is led by Nizar Baylis. We have a study that's led by Luciano Costa of the combination of carfilzomib, Venetoclax, and dexamethasone versus carfilzomib and dexamethasone alone. That study was presented, I want to say, in 2018, and hopefully we'll see some further data later this year about that. That looks like to be a safe and potent combination. There's a large international randomized trial in 1114 only. Again, all these are now 1114 only patients of Venetoclax and dexamethasone. Venetoclax and dexamethasone versus pomalidomide and dexamethasone. So that's an ongoing study. And so all these are ongoing studies. We have investigator-initiated studies here looking at other rational combinations that we're trying to get opened to continue to investigate this.
So can I ask a question? Because
of course.
I've heard other people talk about when you start using more of the immunotherapies that you may not want to use Dex because it could suppress your immune system or whatnot. So I've heard of some combinations trying to kind of get rid of the Dex. but you're saying this is a really great combination for Venetoclax and you should keep it. How does it all play in when you start doing trials like Velcade, or Venetoclax Daradex, kind of?
Yeah, it's a great question. I think because there's a rationale for dexamethasone making Venetoclax better, there's a rationale for having it there, at least in the beginning. But what we try to do, I think once you get past the first, couple of months of treatment, then based on tolerance, rapidly dose reducing the dexamethasone. And because while in the beginning it's probably very important for a response, it's probably less important for maintaining the response. And so I think that's really the key. I don't think we see the same, because we don't think that the reason Venetoclax works is because of activation of the immune system, then I don't think we have those same concerns of using dexamethasone that you would have in a CAR T cell or in a bispecific or something like that.
Yeah, that makes a lot of sense. And it would make sense that you would start combining them with the things that are really well known and well used, like the proteasome inhibitors and the IMiDs. Are there trials as well using them with, you talked about the Velcade Daradex versus Velcade Dara, right? It's
Venetoclax Dara versus Velcade Daradex, right.
Oh, okay. Venetoclax Dara versus, I wrote that down wrong. And then there are other trials too where they're using it like Venetoclax Daradex with or without bortezomib, right?
Yeah, that arm of that study has modified into just Dara Venetoclax Dex at 2 different doses of Venetoclax versus Dara Velcade Dex. We're not looking at we've pulled off from looking at that 4-drug combination at this time.
Oh, got it. Okay. And then it's also being tested with ixazomib, right? So I don't know what the difference is between ixazomib, normal ixazomib, and ixazomib citrate, is it? But that's what the study said. I don't know what the difference is there.
Yeah, that's ixazomib. That's ixazomib. Yeah. So again, just like looking at Venetoclax with Velcade as a proteasome inhibitor, carfilzomib, Kyprolis as a proteasome inhibitor, looking at it in combination with ixazomib or Ninlaro as a proteasome inhibitor, also it makes sense.
And then these are all for, what phase of studies are these? And they are all for relapsed refractory myeloma patients, right? So
right. These are So these are all they're all phase 1, phase 2 studies, and that's not true. They're phase 1, phase 2, and phase 3 studies. The study of Venetoclax dexamethasone versus pomalidomide dexamethasone is a phase 3 study, and that's it's a very large international study. Looking at standard outcomes, including survival, and the other ones are earlier. They're randomized, but they're still earlier because they're still trying to figure out, is this safe and is it good enough to do a phase 3 study?
Right. And how long do you think it will be before? Because if you just have one phase 3, they're still kind of in earlier forms. How long do you think it will be before they start kind of moving it up? And since if things are in phase 2 and some in phase 3, you already have the safety data, right?
Yeah. So I think so far to date, we don't have confirmed safety data from an overall survival perspective that to move it up so the Venetoclax Dara versus Venetoclax Velcade right now could be used as early as second-line therapy. So, but that's as part of a study. And so I think we'll be able to see wider use once we have a randomized study in 1114 only that proves that there's no decrease in survival. That risk that we saw in the all-comers is not there in the selected population.
Yeah, I remember going to that. That was a couple of years ago, right, when they were going through that? So how many 1114 patients are there total in myeloma? Do you develop that translocation at the beginning of your disease, like even in the smoldering stage? Can you develop it over time, or do you pick it up? And well, let's start with that.
Great question. So it's about 15% of patients have the 1114 translocation, and it's early. It is part of the reason that the cell became a myeloma cell from a normal plasma cell. So you can find 1114 translocations in MGUS. You can find it in smoldering, and obviously you can find it in myeloma. And if you don't have 1114 early, then you really shouldn't. You shouldn't pick it up later. It's certainly possible that it wasn't identified early and you only see it later, but that really does. But you want to think about this translocation, like all of the 14 related translocations, as an early event.
So for your patients, I mean, you have a lot of 1114 trials running with these different combinations.
Right.
Do you just encourage your 1114 patients, hey, you should always consider joining one of these trials?
Exactly. That's exactly what we do. I mean, that's the only way that we're going to be able to get Venetoclax to everybody, is to everybody who could benefit from it, that is. When I say everybody, I'm just talking about the selected population of patients. But the only way to get Venetoclax to today's 1114 patients and the 1114 patients in the future is to participate in clinical trials. Yeah.
Do you have any early indication whether, because you were talking about, when it was being used with bortezomib and things like that, and you're not seeing a similar safety, thing for the 1114 patients. Do you have an idea of IMiDs versus proteasome inhibitors, which is going to be a better combination, or do you just go with like standard RVD or something like that, and then add Venetoclax to
right
it?
now I think right now the data we have is mostly with the monoclonal antibodies and proteasome inhibitors, and there's just not a lot of data in combination with the IMiDs yet.
And Venetoclax by itself, do you see any particular side effects that patients need to be aware of for Venetoclax?
Yeah, so, great question. Venetoclax, again, want to be conscientious about infection risk. It can lower blood counts, and it can be associated with some GI toxicity, nausea or diarrhea. The other thing that's really important in Venetoclax is comes from the data that when it was first developed for CLL, and some patients with CLL developed fatal what's called tumor lysis syndrome. The drug killed so many CLL cells at once that all the cells died, and the insides of all those cells came out and then damaged the organs to the patients, and the patients actually died from what's called tumor lysis syndrome. And so now in CLL and other diseases, there's a very, very cautious ramp up. You start at 50 milligrams, then you go to 100, then you go to, or even you start at lower than 50. You start at 20, and then go to 50, and then 100, and so on until you get to the target dose. This is in contrast to myeloma, where while it's still very effective, we have not seen serious tumor lysis syndrome. So in myeloma studies, we either start immediately at 400 or 800 milligrams a day, where in CLL, they start at 20 milligrams a day. We have found, and I have patients that we've treated that we can tell have responded to a single dose of Venetoclax at 400 or 800. We can tell they've responded in 24 hours. I know when I treat a patient if they're responding 1 day later.
Oh, that's incredible. I had no
incredible.
idea. How do you determine
It's
that?
incredible. So we've actually done light chains the next day and seen patients' light chains go down. Patients who have circulating plasma cells that don't quite have plasma leukemia, all their circulating plasma cells go away. Kidney function gets better the next day.
That's incredible.
Yeah.
Yeah, so let's talk about access for a minute.
Okay.
So I had a friend named Steve, and he actually submitted a question for a little bit later, but, he had 1114. Nothing was really working for him, and we had included Venetoclax in HealthTree, so he saw that as an option, went to his general oncologist, and he said, oh, no one would ever give you that drug. And then he thought, well, I should probably go talk to a myeloma specialist. He talked to that specialist, and the specialist said, actually, that would be a great option for you. And ended up getting it and working with the oncologist to convince him that that was a good idea. And he's been doing really well ever since. So how do patients... tricky.
I would say that it's very tricky. So in theory that you could get it, but I think we have to, again, while I've seen really remarkable responses, we don't have the clinical trial data that where we've proven without any doubt that the benefit outweighs the risk. And we have to be honest about that. That's where we are today. Now, obviously, you heard me talk about this, how excited I am about it, but I'm also honest about it and saying, and the FDA is, if we knew that the benefit outweighed the risk, this would be an FDA-approved drug. That's how it works. Yeah. And so, it is extremely rare circumstances that I would consider that. And primarily the circumstances are that the person's not eligible for any trials and not eligible for any other therapy. And we've had a very frank and open discussion about the lack of complete data.
How long do you think some of this data will take to read out? Because if you said that it shows up early in the MGUS and smoldering myeloma stages, you would think that maybe you nip it in the bud at those early precursor conditions by using Venetoclax with Dex or something like that.
Yeah, that's interesting.
Early stages.
We're a long way from there. Great. I mean, it's a great idea. But right now, what we have to focus on is the relapsed refractory patient population with 1114. Let's prove safety. Let's prove that the risk-benefit ratio is positive, and then start asking the wider questions. But we have to just do the first step first, and get the studies done that demonstrate to the world and the FDA that this is a safe and effective medication in patients with 1114.
Right. So if there are 4 or 5 trials running in various stages, how long would you anticipate that? I don't know. I mean, we're looking ahead, right, at things like CAR-T, and we're looking ahead. I'm going to try to say 2 years. Ballpark it. Yeah. It's not fair for it to do that to you, but.
I actually have no idea, but 2 years is my guess.
Okay.
And that is if it proves to be safe. If the risk benefit ratio proves.
Right, and you just need the data before you know that. And this is one really important reason. And so I encourage everybody who has 1114 who is relapsed disease is seek out one of these trials. Call me. Well, call Jenny. She'll call me. We'll get you on study if it's appropriate for you. I think this is a really important point, and we stress this on every program because this is how myeloma patients have the power to move research forward. I mean, it is by joining these studies. and learning. Yeah.
If we
Go
don't
ahead.
do these trials today and tomorrow, and if we don't do these trials in the next year, then 3 years from now, we're going to be having the exact same conversation, and we won't have we won't have the right, and we won't have the drug if it's appropriate.
Yeah, I agree. And just clinical trials in general. I mean, the development in myeloma is happening so fast, and we could come to conclusions even faster if patients really thought about that as a treatment option. And I know in this situation, it's really for relapse refractory patients, but there are open clinical trials for every stage, whether you're, even observational stuff in MGUS and smoldering, and even treatment and smoldering. So I would just encourage all our listeners to think about clinical trials. Every time you make a treatment decision, think about them. And, we have tools in HealthTree, and we're linked to Spark Cures in HealthTree, so you can find clinical trials that you're personally eligible to join. It's not as hard as going on clinicaltrials.gov anymore and just doing this search on 400+ clinical trials. And we can connect people with investigators like yourselves to get people on study. So I highly recommend it for all myeloma patients.
Agreed.
Okay. Well, let's just give a couple minutes if people have any questions. So if you have a question for Dr. Kaufman, you can call 347-637-2631, and then you can press 1 on your keypad. And sometimes people are very shy, but I would encourage you not to be shy and just ask your question. Okay, so we have a caller at 414-4302. Go ahead with your question.
Hello, this is Anthony Link. How are you guys doing today?
Great.
Good.
How are you?
I'm doing good. I've listened to the whole program. I appreciate that. I actually am wanting to get into the trial, but the problem is and I've been talking to my doctor in Cleveland Clinic, I am not in complete remission, but I'm also not where it was progressed enough. But I can tell you that I had a stem cell transplant that the day I left University of Miami, my kappa numbers were higher than before. My my therapeutic drug that I've tried to do just does not put me into complete remission. Thankfully, carfilzomib stopped my going from 21 day to the next week going to 200. But at the same time, I'm not in complete remission. The one thing I don't understand about what you had mentioned, which is that going through a stem cell transplant, going through all of these therapies, my body is going to be worn down. I would much prefer, because I have the 1114, understanding the full risk that with what I'm doing, take the Venetoclax, because what I don't understand about some of the trials is that I have gone now 5 years wearing my body down. And I would prefer if I could have done a trial like this now, a year ago, 2 years ago, instead of keep on going through the chemo, waiting for my numbers to go up, and then I'm even more worn down than before. So I guess I'm trying to understand, why is there not a cohort or something for someone that cannot get into remission? I can never get my Lambda number goes off, my ratio is never off, and I'm willing to do it because I understand the risk, but I'm willing to do it because my body's going to be worn down at the time that they're taking the other patients. I'm trying to understand this going back and forth, and I even think I've emailed with Jennifer, a couple of months ago. There's this cycle that the drug that myeloma patients are going through, and by the time that they progress, then their body can't take it.
Yeah. So I hear what I completely hear what you're saying. the entire structure of how we do trials for approval are all based on patients' myeloma not getting worse versus trying to make myeloma that's okay better. Now, having said that, I think it's a very astute observation, which is if you've done a treatment and you're left with a residual disease, we should be targeting that residual disease in a different way. Totally agree. And those types of trials are not the type of trials that are going to lead to drug approval, but you'll find those types of trials, like at our center, we're currently building that exact trial after treatment, seeing what's left and targeting what's left. It's a great idea. If we ever have that trial that takes 1114 patients and uses Venetoclax to do that, I will let you know, and we'll make sure that's there. But I understand. But your doctor's right. You're not going to be eligible right now, and it's just. And you're right. It's such a challenge to hit that perfect time where you're eligible but you're not too sick. It's a challenge. I think we all agree.
Yeah, that's a great question, and yeah, I love that question. Thank you so much for asking it. Okay, 305-7339. This will be our last question because we're at the top of the hour.
Yeah, hi, this is Victor Taroni. Hi, Jenny.
Hi, Victor.
And I'm actually, I went down to Emory and got tested, and under the recommendation of Dr. Nilka, I'm on 800 of Venetoclax and 20 of Dex, and almost for 2 years now. It's been working pretty well. And so I'm kind of thinking about the future and for me and wondering what is the range of response times that you've seen? You know, how long have people been on this successfully?
We have a patient.
The other question is once it stops working, I've heard pitavastatin is a possible combination. I wonder if you could comment on that.
Go ahead.
Well, and the other thought is, if it also stops working, could you add something like Dara or Kipros to it, or would it be best to just abandon the Venetoclax and move on to something else?
Okay, so three great questions in one. Perfect. So firstly, our longest patient, who is the young woman I talked about, is still on trial and still in complete remission. And I want to say we started in 2004. So that's 6 years. I'm sorry, 2014. It could be 2015. So it's 5 or 6 years. So it can be a long time. Secondly, there's probably, depending on your case, there's a lot of treatment options. I think you were talking about pevonedistat. That's one treatment option, but I think we don't have a
pitavastatin.
I'm not sure what that one is. I might know it by a different name.
It's a standard statin drug.
I can't imagine that a statin drug would work as a single agent.
No. With combining it with the Venetoclax.
Oh, I see what you're saying. So that looks like it's effective in the laboratory. I do not recommend using statins with Venetoclax based on the laboratory data. Based on the laboratory data alone, while it's a good idea in the laboratory, there's no clinical data. If you need a statin for your cholesterol and you're on Venetoclax, great. I would not take a statin unless you needed it for standard reasons. And then the third question was, if what we've learned is when Venetoclax stops working, it stops working. And so adding things into it aren't the right thing to do. The right thing to do is switch.
Okay, that's
great.
That's very helpful. Thank you.
Yeah. Okay. Thanks, Victor. Great questions. Well, Dr. Kaufman, thank you so much for spending the hour with us. We are so grateful. Yeah. I learned a lot about 1114 myeloma today.
All right, you should talk to me more often.
Yeah, I should. It's awesome. And Venetoclax. So thank you just so much for participating. We're just so grateful. There are people like you working on myeloma. It's awesome.
Yeah, no problem. I'm happy to do it. It's my life's work.
Well, we're so happy you've dedicated it to us. and to helping find a cure for even a subset of patients and beyond. So we're just so very grateful. Thank you. And thank you for our listeners for listening to today's episode of Myeloma Crowd Radio. We encourage you to tune in next time to learn more about the latest in myeloma research and what it means for you.
Hello, it is Ryan, and we could all use an extra bright spot in our day, couldn't we? Just to make up for things like sitting in traffic, doing the dishes, counting your steps, you know, all the mundane stuff. That is why I'm such a big fan of Chumba Casino. Chumba Casino has all your favorite social casino-style games that you can play for free anytime, anywhere with daily bonuses. That should brighten your day a little. Actually, a lot. So sign up now at ChumbaCasino.com. That's ChumbaCasino.com.
No purchase necessary. VGW Group. Void where prohibited by law. See terms and conditions. 18+.